Biofilms are communities of attached microorganisms, including bacteria. It's the preferred way that bacteria and other microorganisms like to grow, attached to surfaces and in communities where they're protected from the external environment. Clinically, most infections treated by clinicians involve biofilms, and our interest is in biofilm-associated infections which are associated with implanted medical devices. So, for example, something like catheters, which are made of silicone materials like this, and bacteria that are present on the skin and mucous membrane will like to attach to these implanted biomaterials, forming biofilms. And the biofilms then are very resistant to our own immune responses and indeed to antibiotics and other antimicrobial chemotherapy. In order to make our research more relevant to patients, in investigating the mechanisms through which Staphylococcus aureus and MRSA forms a biofilm on an implanted medical device, we try to recreate what happens in the human body. So we coated our biomaterials with plasma from human blood and then examined the way in which the bacteria could attach to the now coated surface. And what we found was a completely new mechanism of biofilm where the bacteria use the enzyme coagulase to convert fibrinogen into fibrin. And fibrin is something that we know about because it's involved in blood clotting, but in this context the bacteria are using fibrin to stick to each other and to the surface of the catheter, which then gives rise to a very chronic infection that's very difficult to treat. We now know a target in order to disrupt that, and basically the target is fibrin. So if we can use fibrinolytic enzymes or fibrinolytic drugs which disrupt and break down fibrin, that will then disrupt the bacteria from the surface of the catheter, making the bacteria more susceptible to conventional antibiotics and antimicrobial drugs, which could hopefully lead to a new type of therapy for this type of infection.